All right, looks like we are live, and welcome to Standing for Truth. I am your host, Donnie. Thank you, everybody, for being here for tonight's epic debate. What are we debating tonight? We are debating abiogenesis. This is the much anticipated debate on abiogenesis between Tony and Luca, the Boston Brawler, also known as the creationist Ben Affleck himself, and the Italian wonder, the man with the plan, Luca Medugno. Gentlemen, Tony and Luca, thank you for being here for tonight's important debate.
Okay, I can start my presentation. my yeah,
No,
no,
not yet,
my
not yet, Luca.
introduction.
Not
I'm
just yet. You're
Luca.
getting too
I
ahead of
think
yourself,
that
Luca. I just wanted
everybody,
to
yes,
Yeah, we'll do intros just in a second. I just want to remind everybody if you're not yet subscribed, please make sure to hit that subscribe button. And also in the description box, you'll find our websites and you'll also find our Patreon where you can help support Standing for Truth Ministries. Now, this is day 4 of epic debates this week and shows in general. At the beginning of the week, we had Russ Miller here for a presentation on the Grand Canyon and the Grand Staircase. On Tuesday, we had Dr. Mark Armitage here. We discussed dinosaur soft tissue. That one was definitely one to remember. Please check that out. Last night, we had the epic debate between Dr. Dino and Wade the Wizard. Kent's first time debating a wizard. So that was definitely an epic debate. We had over 320 people in the chats getting great feedback. Please check that out. And now tonight, the great abiogenesis debate, Tony Maurice and Luca Medugno. And guys, the fun continues next week as well. We're going to take tomorrow off because we need at least one day off here. But next week, we start on Monday with Michael J. Ord. Michael J. Ord will be back here with us for a presentation and lecture on mammoths. What happened to the mammoths? Then the same week, the much anticipated debate between CJ Cox and Dr. Leighton Flowers, the total inability debate. This one is going to be epic. And again, the evolution debate challenge continues next Friday. Mark Drysdale and Kent Hovind, they will be debating the topic of evolution. Okay, enough for me. You guys didn't come here to see me. You came here to see the superstars for the night, Tony, Luca. Let's start off with some introductions now. And Luca, let's start with you, the Italian wonder. What's going on? How you been? A little bit about
yeah,
yourself. Go ahead.
yeah. Sorry for before, and yeah, I am a seasoned debater. Seems to be it, but yeah, you all know me. I am a chemist. I have a degree in chemistry. As for now, I'm working as a teacher in an elementary school, and I'm very passionate about this kind of debates. This night, I'm particularly excited about this debate because I'm debating Tony. We did debate this same topic 2 years ago, so I'm really pumped for this debate. I cannot wait to face Tony another time. So yeah, this time, this night will be fun.
I appreciate that introduction. Luca, yes, you are quite seasoned now. You've debated Kent Hovind himself on this channel. You've debated Nephilim Free, Otangelo, Grasso, now Tony, and also you'll be debating me next month on is there evidence for human evolution. And Tony, the man himself, I appreciate you being here for this important debate. I think this is it's not your first time on the channel. You've been here before helping me interview people like Dr. Gordon Wilson, Dr. Ryan Hayes. You've interviewed Dr. James Tour. So, you are well known here in the community. But Tony, I think this is your first formal debate on the platform. So go ahead, introduction. Who are you? What's going on? And go ahead, brother.
Yeah, thanks for having me. Tonight I'll be making a case for chemist interference, designed equipment, and some of the unrealistic lab environments. Not claiming to be an expert in the field, but hopefully it'll be a great discussion, and I appreciate it.
Absolutely. I appreciate that, Tony. I appreciate that, Luca. So for the audience sake, I want to go over the debate format for the night. So again, it's going to be a formal debate. We're going to be having 12-minute opening statements. Luca will be starting us off there. Then we're going to be having 8-minute uninterrupted rebuttals. Then we're going to have a discussion portion, about 30 minutes, where the debaters will be asking each other questions pertaining to the topic of tonight's debate, which is abiogenesis. And then we're going to have 5-minute closing statements. And then this is where we get you guys in the audience involved. We're going to have an audience Q&A. So please make sure that you are tagging me with your questions at Standing for Truth. And that way I don't miss them. Then right after the debate, we're going to have another epic after show over on Logical, Plausible, Probables channel. So the debate continues over on John's channel. Sounds like it's going to be another all-nighter, just like last night. So that being said, Luca, we're going to hand it over to you. But real quick, Luca, a couple people in the chat saying that your volume's a little low. So from my end, I am going to, especially for your opening statement here, I'm going to increase your volume a bit. Let me see. Let's do an audio check real quick before you start, Luca.
Okay. I'm fine. Everybody can hear me fine.
That sounds good for me. What about you, Tony?
Yes. I don't know.
Yeah, you can hear me good?
Yeah. Oh, yeah.
Yes.
Okay. Yeah, Luca, so I'm going to increase your volume for your opening statement. I want to make sure people are hearing your arguments and points, and we look good. So, Luca, whenever you're ready, floor is yours. You got 12 minutes. If you need to share a screen or anything, let me know, and I can do that for you as well.
Yes, I need to share. Okay. Allora. Okay. You can see it?
Yes.
Okay. I can start. So
go ahead. Floor is yours.
Okay. Abiogenesis debate. Is the hypothesis useful? I think so. Here you all can see how the field of origin of life is quite active. Here you can see my Google Scholar research tab. It's in Italian, so I did put in the translation. And you can see how many papers they were published only by this year, 2022. Almost 7,000 papers, a lot. Of course, quantity is not quality, but why it's important, this topic, why abiogenesis is important. Yes, it's important because we can understand, we can know a lot of things. Much of these discoveries are really, really important, and some of these processes can help us to understand better some very important mechanisms, like autocatalytic reaction, how to achieve homochirality in certain ways and in certain situations. And yes, prebiotic chemistry in general, it's very important. Here you can see some papers that can support my point, very important things achieved or that scientists are trying to achieve. So polymers, homochirality, autocatalysis. I chose one paper for each thing. I think that those things are important, and it's important to continue to study this topic. So the research is done just in the lab or elsewhere. As you can see, no, it's done outside the lab also. This researcher is called David Dreamer. Also, there is another researcher helping him, Vladimir. I will not say his last name, I will butcher it. So this is Dreamer doing an experiment. Here you can find a paper, a couple of paper actually, that discuss what these people are doing. They are trying to reproduce the environment for this kind of reaction and the prebiotic chemistry at large in the field. So it's a very fascinating topic. I think that people should read about these things. And also, I'm not talking about the moon of Saturn, Titan, and it's very important. You can see there why. Titan is one of the few places in our solar system where life could in theory develop outside of our planet. And the Cassini-Huygens mission has confirmed the presence of several organic compounds on the atmosphere of this moon. So it's a very important discovery because you can relate with abiogenesis and all of these topics. Maybe we will find more about this place and we will find something interesting there. Maybe even a new interaction of abiogenesis. Why not? For now, we just know that there are some kind of compounds that can lead to life, but we still do not know a lot about this. So that's a favorite of mine, homochirality. It's a lethal blow to abiogenesis or not? And we will discuss about the topic for the next slides. So, a brief introduction to homochirality, because I do not think that everyone in the public can know this kind of things. Homochirality is an uniformation of chirality. Here you all can see what a chiral molecule is. Basically, molecules with the same elements in it and same chemical and physical properties, but molecules that cannot be superimposed. To talk in a more layman way, we can say like your hands, your right and left hand, to understand a little what those compounds are. Why it's important to talk about these compounds? Because life does show that you can see typically a left and a right hand configuration, and in life we will find some very peculiar things. So why it's a problem for abiogenesis? Because homochirality is observed in life. Amino acids are almost always left-handed, and sugar at the opposite are right-handed. And we never found an example for something different, and can see it can be that can be a problem for abiogenesis at large. Of course, we need to explain why we find this kind of things. So scientists are putting up some hypotheses on how to solve this problem. We will look into two of them: liquid chromatography and crystal formation with symmetry breaking. So yeah, here you can see how chromatography works. You have a mobile phase that passes through a stationary phase, in this case liquid and liquid, and different compounds will react in a different way, and they will move a different velocity, and you can sort of these compounds with this process. Okay, here there is a very important paper to me. This paper show how can you get to homochirality with only liquid chromatography. Basically, they took some liquid organic compounds, and they put them in a beaker or a reactor of some kind, and they observed that do some time they did sort themselves in various domains, and in these domains homochirality was achieved. You can see the experiment, the racemic solution at first, and then with the passing of time, the formation of domains with homochirality in it. Of course, this process is relevant to abiogenesis because you can get with this to homochirality, and homochirality can lead to life with only one stereoisomer in it. It's very important. Here you can find the paper. It's quite a good reading, and I will talk about it in detail. Okay, symmetry breaking with crystal formation. You can crystallize a racemic mixture, and those crystals can and do melt a different temperature from the racemic crystal, and this can lead to a selection, sorting, and you can sort out this kind of compounds with this method. So it's possible to create life. We talk about protocells. Protocells are structures that are similar to cells, but not quite cells, and you can find it in nature. And we actually can create them in the lab. It's very important. And they show some of the behaviors of life. They are not yet alive, but we can get quite close. They show those artificial protocells predatory behavior, and even they can develop genes inside the structure. They can be used as basically micro bioreactors. So, conclusion, abiogenesis not at the end. Dozens of papers are published each year. Researching on this topic can lead to new technologies. Perceived problems for the hypothesis can be solved in due time. It's possible to create life or something similar in the lab. And the hypothesis for me is plausible. Here you can see my degrees, relevant ones, my thesis, and thanks for watching. Sorry, I did use a little bit more time.
No worries at all, Luca. We're a little more easygoing.
I'm no longer putting up my screen.
Nope. You're good. Okay. You're good. So it's just us on the screen now. Time flies by, Luca. I appreciate the opening statement. Clear, concise. I appreciate the visuals. And we're a little more easygoing on the opening statement. So that was between 12 and 13 minutes. And so, Tony, we're going to give you equal time, my good man. So you have between 12 and 13 minutes for your opening statement, Tony, whenever you're ready.
All right, let's go. Well, are scientists showing life forming from purely bottom-up natural processes, or guided chemistry and intelligent design? Science works from what we do know, not what we don't know. So what have we learned in over 130 years of origin of life experiments? Well, there were no discernible sources of naturally occurring homochiral amino acids to polymerize into proteins. No place to buy purified nucleotides, nowhere to order homochiral sugars, no stores that sell phospholipids, no human-designed coupling agents or protecting groups, no human-designed pipettes to sequence very specific purified bases, no pristine glass vessels, no purified solvents, no degassing steps, no vacuum pumps, no ability to conveniently turn reactions on and off, no highly calibrated design sequencing machines, no purifying chemical solvents in high concentrations to remove impurities and other contaminants, no simple method to transfer chemicals from one reaction vessel to the next and eliminate toxic tar buildup and other poisonous byproducts. No plexiglass incubators to hold chemical reagents at just the right temperatures and just the right pH. And of course, no intelligent human chemists. Early Earth did not have that luxury. Time is always claimed to be the great savior of abiogenesis. Time is actually the enemy. Carbohydrates and amino acids are constantly dispersing and caramelizing. A prebiotic system does not have the ability to purify its own ingredients. Donnie, if you want to throw up that HPLC machine, give you an example of what liquid chromatography is. Okay, this is a high-performance liquid chromatography machine. This is what Luca is proposing as a possible solution for the ongoing chirality problem. The problem is these machines run off of benzene, purified water, tetrachloride, and other chemical solvents not readily available all over the early Earth. You can see there's different chambers there, vacuum pump system. You have to move molecules in a direction that you want them to go in. And of course, it's operated by an intelligent chemist. Now, I'm not saying this machine would be available on the early Earth, but you would have to have a series of chemical puddles or a natural mechanism that is able to accomplish what this machine is doing. You would have to have one puddle purifying your amino acids, the next puddle your nucleotides, and then another puddle purifying some sort of membrane, lipid membrane, and it's all going to come together somehow. It would be a worst case for a chemical ocean. Your molecules would be dispersed, and just purely natural processes would not be able to achieve that. Luca has brought up the crystallization problem. Because the physical properties of enantiomers are identical, they seldom can be separated by simple physical methods, such as crystallization. Louis Pasteur experimented with tartaric acid. It's a derivative salt. Pasteur noticed that crystals of tartrates had small faces. He then observed that in racemic mixtures of tartrates, half of these crystals were, of course, right-handed and left-handed. and this is the part I wanted to emphasize, he painstakingly manually separated them. Now, I'll agree with Luca that we can manipulate yields in the lab, but it's always due to intelligent interference. Now, Luca's probably saying, well, you're just biased, Tony. You're not really studying the chemistry and what the scientists are saying. Well, what are some of the scientists saying about the chemistry that actually do agree with you, Luca? The improbability of natural DNA RNA formation. It would be a miracle if a strand of RNA ever appeared on the primitive Earth. Leslie Orgel, he was actually an atheist. You would have to build straw man upon straw man and presuppose to get to the point where RNA is a viable first biomolecule. Gerald Joyce, agnostic. RNA is harder to form than proteins and amino acids. No nucleotides form from plausible Miller-Urey type experiments. RNA is hard to form the molecule's backbone due to rareness of phosphates, 0.08 parts per million in a vast chemical ocean. Very dilute amounts of these chemicals are going to be able to concentrate. Inability to produce ribose without hundreds of other sugars present. Acceptance of only right-handed ribose. Ribose in a solution always has a half-life of 1 to 2 hours. Again, your molecules are constantly breaking down and dispersing, whether this is a laboratory environment or an outdoor natural environment. Water breaks down nucleic acids, hydrolysis, molecules constantly dispersing. UV rays and metabolism byproducts cause lesions. Hard to form nucleotides. 2 bases only form near freezing temperatures. Other 2 bases can only form near boiling temperatures. Vast temperature changes all across the primitive Earth. How would this be controlled? Hard to chain together components without other chemicals mixing in. Origin of life experiments always follow protocols to this. Purchase chemicals in high purity from a chemical company. Mix those chemicals together in high concentrations or in a specific order under some set of carefully devised conditions in a modern laboratory under a high level of control. And I'm going to close now with Harold Urey, spent his whole life in the field. What did he have to say after 40 years of research? All of us who study the origin of life find that the more we look into it, the more we feel it is too complex to have evolved anywhere. We all believe as an article of faith that life evolved from dead matter on this planet. It is just that its complexity is so great, it is hard for us to imagine that it did. Can yield my time for now.
All right. I appreciate that opening statement there, Tony. Clear and concise as well. So fantastic job from the both of you. And chat is great. Chat is lively. I want to remind you guys in the chat, please tag me with your questions at Standing for Truth. And as always, we'll have a fun audience Q&A. So Luca, we're going to hand it over to you. We're now moving into the rebuttal portion of the debate. We have 8 minutes on the clock. I'll give you a 1-minute warning, just so you know to start winding things down. And Luca, you are on mute. Just make sure you unmute yourself whenever you're ready.
Okay, I just have some questions and problems with what Tony said. So first of all, what I propose as liquid chromatography is nothing like high performance chromatography. It's just 2 enantiomers interacting with solvents, and that's all it is. It's possible to have that on an early Earth. Yes, it is. It's just a liquid inside another liquid. It's all that is. And all those things, autocatalytic reaction, liquid chromatography as specified here, crystal sorting with enantiomeric sorting of those crystals are things discovered and used basically in the last decade. So yeah, there are pretty new things. Even more recent than that, because if we are talking about liquid chromatography, we are talking about a paper about 2020, 2 years ago, and that paper it's quite good, and I want to talk about that in detail. Also, RNA, yeah, we cannot have RNA. RNA just need clay, some sort of clay, and a cycle of drying and be wet again, and so on and so forth. They are trying to reproduce this kind of process in volcanic puddle. You saw that scientists doing so, and it's not that far-fetched. Also, RNA and DNA are not the only nucleic acid you can have. The first cell, the first living thing will not be what we think about life now. In one of the papers cited by me, they basically they got these protocells with an inorganic structure protecting the inner cell. So it's possible to get something like a cell with crystals, basically silica crystals and structures. So that's a possibility, and scientists are investigating about that. So also the moon of Saturn, Titan, we can look at it and see all those organic compounds there. So we do know that something like that, the synthesis of those compounds, is possible as elsewhere. So we can learn a lot from that. And Titan, it's not been stained by scientists. It's no one ever got there, so we can observe from afar, and it's quite important. I would love to discuss more on the lab thing, how chemists work. It's not like we are doing magic. We are using to our advantage chemical process, and it's not like it's impossible to have those in nature. So yeah, people are trying to get to the pathway of life with computer simulation. That's the first step. You need to have a lot of interaction to find a viable pathway, and in the lab and in the field, it's quite hard to do so. But we are getting there. There are some models do exist right now. I do not trust those models because no computer is good enough to reproduce reality. It's impossible to do so, but they can get close. From there, you just need to reproduce that pathway or those pathways, and I think chemists they will find a way sooner or later, and I don't think that it will be too much in the future. And that's it. I do not think that abiogenesis is impossible, far from it. I rest my case.
All right, I appreciate that, Luca. With about a minute to spare, that we can toss into the open discussion portion. So, that being said, we're now going to hand it over to Tony Tony, you now have your 8-minute rebuttal as well. So whenever you're ready, the floor is yours.
I would say every chemical environment you're mentioning, Luca, is always going to be moot and uncertain. These chemists are guessing. Even the top researchers are admitting that. You've said that we don't know, in other debates that we've had and discussions, they don't know the exact environments of the early Earth, but it's so funny how chemists will put into their experiments exactly what they want and get the exact results they want. How convenient, right? I think it goes far beyond just investigating the process. You mentioned protocells. Protocells are not lifelike. They're nowhere near life. The requisite ribosomes, mRNA, and enzymes, reagents were purchased. They're always purchased from chemical companies, and they're always extracted from pre-existing living systems. Really, it's just more materialism of the gaps type arguments. We're going to figure it out one day type of argument. It seems that's all that the YouTube atheist is presenting. Yeah, I can yield for now.
All right, Tony, short and sweet. You ended in the middle of a sip of coffee. So there we go. We will restart the timer and move into the open discussion. Gentlemen, we have a ton of great points to discuss. Luca and Tony, as you guys are having your discussion, I will be saving questions. I see questions coming in as well for the audience Q&A. And as always, we like to keep this as equally timed as possible and focusing on one topic at a time. So since Tony, you just ended with your rebuttal, Luca, let's hand it over to you to kind of kick us off with this discussion. Go ahead, gentlemen. The floor is yours.
I'm pretty curious about something. Tony, you said that homochirality was one of your biggest problem with abiogenesis, and I do think that those two methods presented here are quite believable. Crystal sorting does work quite well. It was discovered more or less 10 years ago. They are still improving on that thing, but it does work. Liquid chromatography too, and it's relevant to the topic because it was liquid inside a liquid, and the proposed environment for abiogenesis is an aqueous one. So what do you think about that?
What were the exact starting materials, I would like to know, and what's the process that is actually used? You're saying you're not using an HPLC machine. This is another type of chromatography. I don't know every single method.
Another type. Liquid chromatography does not need a machine. You need a machine to have high performance, but chromatography, you can have it with a drop of wine and a little bit of paper. That's chromatography. It's not like you need a very complex machine to do it.
And you're getting a full separation in yield?
Sorry?
I would be curious if you're getting all left-handed amino acids and all right-handed sugars. That's exactly what it's doing? And what are the exact...
It is. I've shown you the picture. It's sorting into domain of different enantiomers. You just need a racemic to be inside a liquid, and they just need to interact with the solvent in a certain way to sort themselves into...
That's why I would be skeptical of the actual... Yeah, I would be skeptical of the actual solvents they're using and the methods that they're actually using. You also mentioned
you can find the materials inside the paper.
Sure.
You got inside my presentation some of those compounds,
sure.
the enantiomers at least. They are just organic compounds, nothing too fancy. There are quite long chain compounds, but other than that, nothing too fancy.
Sure. Yeah, I'd like to read them and run them by a chemist.
Yeah.
You mentioned
by a chemist, but the process it's a physical process. Chromatography, it's not even a reaction. It's just a physical process.
And it's sorting all your amino acids naturally.
No, there are not amino acids, but it's sorting through enantiomers.
Right.
On the amino acid, I think that the crystal thing works better.
Yeah.
And that's the good one, because the crystal can sort themselves. Because if you have a selecting factor, like in the paper, a different
I would be skeptical of the actual method.
A different melting point, or a different interaction at some pH. You can have a sorting, because you can have some crystal already solved and taken away by the liquid, and some other still there. And at the end, you will get homochirality with this simple process, and you do not need a chemist to do it, basically.
I'm not sure about that. I don't think chirality is solved by that method. It might be a possible solution. I don't know if it's actually solved. You did mention the clay problem.
Mm-hmm.
RNA forming on clay. There's no problems with that as far as the clay being chemically prepared. Any sort of chemical stabilizers that are used on this clay? Because that, from what I'm reading, that clay is purchased from a chemical company. It's carefully shipped, and it's, you know, in a chemical bath that is not going to fall apart. Your clay is all prepared beforehand. I don't think that's anything you're going to find all over the prebiotic earth.
Sorry, but they are reproducing that environment in real puddle, and it's working for what I know.
Yeah,
you need to understand that, of course, chemists will use already purchased material. You cannot have the time to produce everything from step 1 to step 2000. Of course, you will do experiment on precise steps, and you need the material to do it. But they are finding out ways to get there in the field. They are doing experiment on the field. You saw that in the picture.
Yeah, I would like to know the details of that, if you can.
Yeah, I sent you the papers. I think that only paper I did not send you was the one about the protocells, and I will send it to you. Sorry, I just forgot to add that paper. But
look at what
you already got it.
Yeah, I didn't check it. He must have just sent it to me. But Luca, what's funny is how they're conducting abiogenesis experiments out in the field. I always heard for years that, oh, well, we could never have these types of experiments today out in the field. But I think now, but
was wrong. We are in the field.
Oh, you were wrong. People were wrong because they never wanted to conduct these experiments today out in our atmosphere today. I think they're doing that because they're realizing lab environments are so controlled.
Why it's wrong to reproduce an environment in the field?
Nobody was doing.
You can have oxygen, and that can lead to some problems, but you can find even today on Earth places where this is not a problem, or you can set a tent, pump out all the oxygen, or do something like that and reproduce the environment just like that. It's not like it's impossible to change a little bit of atmosphere. Most of our atmosphere, it's not oxygen to begin with, so you need to make disappear the 20% of oxygen in the atmosphere. No big deal, I think.
And what about life already being here and contaminating your experiment? You're pouring these
You
chemicals into a dirt puddle
take a
out in the
puddle.
field.
Well,
Yeah.
here, just one second. I just want to make sure there's no crosstalk because I just want to make
Yes,
sure
sorry.
everyone's hearing Tony's question. So, Tony, repeat your question, and then Luca will give you a chance to respond. Go ahead, Tony.
What's being conducted actually out in the field? They're just dumping chemicals. There's no other contaminants. There's already life present on this earth. I know that was a big problem for your community that, we really can't conduct these experiments out in an atmosphere today with bacteria, with life already here.
Go ahead.
Okay, if you are conducting an experiment on a small puddle, you will check for life and contaminants, of course. It's not like you will just throw anything in a small puddle and call it a day. It's not how things work.
But yeah, well, that's how abiogenesis works. You have to throw everything all in the puddle and it has to react and turn into some sort of primitive cell. You don't have the luxury of purifying everything that you want and putting in exactly what you want. But I'm sure you would disagree.
No, I do agree, but
you do agree.
Sorry. You just need to find a scenario where those things can add up. Autocatalysis is a very big thing. We are discovering it in the last decade or so. So it's not like we are very knowledgeable about it, but it's a thing, and it's not like it's impossible to get.
Would you say they're investigating the process to actually get the chemistry to work and succeed using an intelligent agent?
No.
Autocatalysis
right. Does not work like that. The autocatalysis is a reaction that, when started, it will feed itself and does not require intelligence to do so. You can find the process. You can hypothesize, put up an hypothesis on how we got there. Of course, abiogenesis is a hard nut to crack because you cannot look at fossils and things like that. And even if you find a perfect pathway to life, even then you will never be sure that's what happened back then, because you cannot observe that. You can just do an hypothesis.
So you're not really skeptical?
Sorry.
Go ahead.
No. I think that we will, and I do not think that it's too far that we will get eventually to a complete pathway. The computer simulation thing, it's a very useful tool in that, because you can set a lot of process, a lot of things in it, and you can rule out a lot of things and find possible pathways. From there, you can start doing experiments and find out the ones that work out.
And you're not skeptical of any processes that they're actually conducting? Are you skeptical?
Why I should be skeptical about those? It's not like they are telling us that all solved, we know everything, and so on and so forth. They are doing a good job. I'm skeptical about the computer simulation, because you cannot simulate reality very well. Not at that level. Chemistry, it's very hard to predict. But yeah, it's useful to rule out the more improbable things, I think. Then you need to do experiments. Computer can get you just that far.
Okay. You wouldn't be skeptical of the protocell experiments? They aren't alive.
No. There is a very important point.
What's being passed off as life?
1 minute. Protocells does exist outside the lab. It's not like we did invent them. We can produce them, but you can find in nature too. It's not just an artificial thing. It's not just made by chemists. And it's a very curious thing, because if life just came here from nothing, the Nilo, so why there are things that are not alive or dead, and virus are the most famous ones, but we got more live.
I don't think we have that intermediate between non-life or something. I don't think we do. It is a protocell. That's what you believe?
Yes, they do. There is an entire field of life with non-cellular life. So yes, you can find it.
But it's not alive, Luca. And what's being inserted into these membranes are actually
It's
pre-existing
not
code.
alive, but it's not dead either. If they can express genes, if they can reproduce, if they can show predatory behavior, what those
Are they reproducing? Are they metabolizing? What's being inserted inside them?
Yeah, gene production, it's a part of metabolism. So yeah, they are doing things. You cannot call them completely alive, because life, for what we call it life, those things are far simpler than that. But they are doing things that only living things do. So they are in the middle.
I would strongly disagree. They're not alive. They're not in the middle. There is no middle ground. And I think you're always going to go to some hypothetical primitive cell that we've never actually demonstrated.
Yeah, except the ones I've shown you that we can do in the lab, that can use silica structure to survive and do things. It's not like they can survive on their own forever. They can do it for some hours, for what I can get from the paper, but we are getting better at it. And it's quite an interesting thing to get something similar to a cell with inorganic outer shell. It's a very interesting thing.
So in a protected lab environment, these cells that's the only place they could exist.
No. Protocells can exist elsewhere. It's not like just.
But they're not life.
Of course, the ones we are making need
Right.
a lab environment, because it's not like we are so good right now, but they can do things. They can do a gene expression. They can do smaller process. So yeah, that's something. And it's interesting to have this kind of structure with inorganic outer shell and the inside working. You can do an hypothesis about that and say, maybe we started life with something like that, with inorganic outer shell. And that's one of the hypotheses, and it's a good one if you ask me. Then much later you can add the biolipidic layer and so on and so forth.
Yeah, that just sounds like a lot of guessing and speculation and materialism of the gaps as usual, Luca.
Well,
you have
the process cells. We did it. And if we can do it in a lab, why we cannot get those?
I think that's the biggest disconnect that the YouTube atheist has. If we can do something in the lab, it's going to eventually happen in nature. I mean, I have a chainsaw that can cut down trees and make lumber, but it says nothing on how that would happen naturally without the chainsaw and the intelligent agent operating it. I don't think protocells are an intermediate. They're definitely not alive, and they're not on their way to becoming alive. And all the ingredients inside of them are all from pre-existing living sources. I couldn't give you the exact chemical pathways, but there is strong evidence that these experiments aren't going anywhere, and they do require pre-existing donor cells to get them going.
Well, it's the most simple thing to do. It's not like you will start from scratch. How many money
But
you
aren't you
think
supposed to?
chemists have?
I think that's the whole point.
Yeah, at the end.
Wait a
yes.
minute. We are supposed to start from scratch. You agree?
No.
You don't agree.
So, first of all, if we look on Titan, we can find, as someone was asking, some sort of precursor of amino acid and so on and so forth. So we can start from there, because we do know that those kind of things can be synthesized even outside our planet, or we need to start from carbon. I
You're
do
talking
not think so. I think we can start a little
You're talking
bit
about the
later.
meteorites that landed here?
No. I was talking about
Crystals?
Titan and Moon.
Okay. I
So
think that would be an even bigger problem to
start from
spread out all
scratch.
your Yeah, I think you would have a bigger problem of spreading out all your molecules all throughout the universe and other planets or meteorites. I still don't think you're going to be able to generate code doing that, but I know you have
It's
wishful
not like
thinking.
that. My argument is, we do know that some of this kind of synthesis can happen even on Titan. Different atmosphere, different moon, okay, but we do know that those processes can lead there. So it's not like you need to start from scratch, like from carbon. Also, if you are doing an experiment, what will you do? Okay, I'll start from a block of graphite and go from there. No, it's absurd. You will try to do experiment on the various steps. Like, I need to figure out how homochirality got there. So you will start from somewhere, not just from scratch. It
I would have to
doesn't
question
make
the level
any
of.
sense to do
Yeah,
so.
I think you do have to start from scratch, from a batch of chemicals, a pool of chemicals. That's what abiogenesis is. If you want to interfere, I mean, I would say that's exactly what I've been arguing is an intelligent interference in these types of experiments.
When
And
you
people even in your field actually are saying the same thing. I don't know why Leslie Orgel would say that it would be a miracle if a strand of RNA would appear on the early Earth.
I
And
do not think
he was an
that
agnostic or an atheist.
First of all, I said that RNA and DNA are not the only nucleic acid out there. So we do not know what were the first one.
And the
Now
hydrolysis
we
problem wouldn't be a
base.
problem.
Why?
I mean, that wouldn't be a problem.
Why?
I'm asking you in
Once
water,
you get
molecules
the
are constantly dispersing, breaking down, caramelizing. It wouldn't be a problem in water.
water is not a problem as you think, because you just need to produce them faster than they are breaking up. My favorite example are proteins. Proteins do solve in water. Everything works out. Thermodynamics, no problem there. The reaction is fine. But the problem is, proteins do not solve in water as they should, because you can take a shower and you will not melt like heated gelato. You will not, simply. Why? Because chemistry is complex. Compounds like that can last long enough, I think. And if you have a cycle of drying and wetting with RNA on some specific clays, of course, you will get those strands. And they are trying to reproduce that environment on the field.
Yeah.
It's not like it's a simple thing, of course, but I do not think that it's that bad that you cannot say, yeah, it's impossible. First of all, we do not know everything about chemistry, but I think we are getting close. Homochirality seemed impossible at first, but now we do know a lot of process that strongly point in that direction. The crystal breaking symmetry, very good one. Very simple one. It works quite well. So yeah, and that's in the last decade. When I was doing my high school years, it was a problem. Now, less so. Liquid chromatography, that's huge. It's quite a huge thing what they discover, the process, because it was not an obvious, very simple thing to think. It was quite a surprise, I can say, that you can leave alone those compounds and get to homochirality.
I don't think anybody's doing it.
Simple, but the process is there.
Yeah, I think you've convinced yourself, but I don't think anyone's actually accomplishing this problem.
Yeah, but they are doing so.
Yeah, they've been running the same experiments, but not really accomplishing it.
I do disagree strongly with that. I think that the field has done very decisive, very important steps in the last years. So, and I'm reading about it, 7,000 papers on the topic. You think that those people are just talking about the nothing?
Well, they do want their grant money every year, so yeah, they have to run experiments. They have to be funded by some people somewhere. Their universities have to function. Yeah, their research facilities depend on funding. Lots of papers on origin of life have been fully debunked or retracted. Jack Shostak did retract his paper in 2018. They can publish thousands of papers every year. That doesn't mean it's going anywhere, Luca. But definitely send me those. I do want to read them.
I already did. But I would love to see you try to disprove those. I think that those are very good papers. So I did read them, and I think that's a very good job. They are doing a very good job. That's it. But okay, some people retracted their papers. I do not know anything about it. I'm not informed about this subject. I would love to know more, but still we do have 7,000 papers published. And I do know people in the research field, and they are not making any money out of it. I don't know about America, but I do know about Italy, and people doing research here, they are poor. I as a teacher, I gain much more than them.
Well, there's $10 million in it for you. Look if you can produce a code. So you could be the first team.
I would love to, but I'm not good enough to do so. Maybe someone will get those money.
Yeah.
I
don't think anybody is, but we can come back.
We don't know. I think they are pretty close to do so. So yeah.
Yeah, we've been hearing that since 1953. They're so close. Every year.
7 decades. What are 7 decades? 7 decades
what
ago,
are seven?
we did
Well,
not have computers. We did not
and look
have
at all the advancements, though. But Luca, look at all the advancements in every other field of science, and look at abiogenesis.
It's not like that, Tony. It's not like science will improve and given velocity. It does not work like that. You will have big breakthrough and huge jumps forward, but you also
I'd like to
will
see
have
that.
long
I think
periods of stagnation in every field. Like we did have huge steps.
I think we've had steady progress in all fields of science. We've been to the moon and back. Look at heart transplants, biology, chemistry, geology. No, I would say all these fields are advancing greatly. Abiogenesis is still in the same sad state it was in 70 years ago.
Except it's not, because they're talking about a lot of things here very interesting, and they are about abiogenesis. It's not like it's nothing. Autocatalytic reaction, that's huge and that's new. It's not like it's something that was known before.
Yeah, I would
We
be
are talking
skeptical of some of
about
the
those
solvents they're
kind of
using.
things in the last decades.
Right. Yeah, I would still be skeptical
And
of some of
yes,
the
it's
conditions,
nothing.
and also the clays that you've mentioned with the RNA world. They're not going to be found all over the early Earth. But I'll come back with some
Different
information
topic.
on that.
If you look into autocatalysis, we are doing some breakthrough right now in these years, and it's a new thing. It's not like it was there before. So you cannot say that this field is dead and it's producing nothing. If that thing was true, no one would spend money on it. Let's be real.
Well, you just said they're not funding their experiments, but now they are. Spend money on
No,
what?
funding experiments, yes. I'm just telling you that the researcher, I do not know about USA, but I do know about Italy, and people working in the field they are not making much money at all. I do more with my teacher job. So yeah, they are doing it for mere passion, not for money. And yes,
No.
funding, yes, you
Yeah.
can fund it, but they will go into materials, experiments, things like that. It's not like people will make money with it.
No, I never meant that. I just meant that the funding behind it. But I
All
would
right, let
say
me
it's
ju-
going to suffer. Go ahead.
No. After you finish your sentence, Tony, we're going to end it there. And Luca, for your response, we'll save it for your concluding statement. So,
Okay.
Tony, go ahead, finish your sentence. I
Yeah,
apologize.
we can come back and do it again and study those specific areas that Luke is talking about. But I would say it's probably suffering from all the same classic origin of life problems.
All right. Well, I appreciate the sophisticated and intelligent discussion, Tony and Luca. That discussion portion flew by. We got some good questions. We still have a solid chat. Also, guys, thank you for making it easy to moderate. Very cordial debate. So what we're going to do now is go into 5-minute concluding statements. So this is your chance, Luca, Tony, if there's anything that you feel was left unaddressed or any final points you wanted to make, now would be the time. We got 5 minutes for conclusions. Luca, we're going to hand it over to you for you to start us off. And whenever you're ready.
I'll just say that abiogenesis is making progress, and even some good ones. So I do not think that this field is dead in the water. Far from it. I think we do live in a very good time for these kind of things. And I just was blown away by those paper I was reading, because I did prepare myself in the last couple of weeks. So I did read a lot about these people, and I was very fascinated by what I was reading. Abiogenesis is not a theory; it's still an hypothesis, and I think it will remain so for a long time, because it's not that easy to get there, because we do not have any fossil, any actual traces for it. We can just move blindly and with a lot of intelligence, trying to find a pathway to life. And I think we will, sooner or later. And I'm pretty hopeful about that, for what I could read. And I cannot wait to get to the questions, because I do want to engage in this topic more.
All right, Luca, I appreciate that concluding statement. And definitely, let's get some more abiogenesis debates for 2022. I think it's an important topic. And so that goes for anybody in the chat, anybody who's watching this debate. Check our website, standingfortruthministries.com, and shoot me an email if you have any debate ideas, any specific topics you would be interested in debating. Yeah. We've hosted now almost 160 debates. So if you're a debate addict, like probably all of us here on the panel,
yes,
check out the website, and that'll keep you busy forever. So Tony, we're going to hand it over to you for your 5-minute concluding statement. Whenever you're ready, the floor is yours.
Yeah, good talk. We can definitely come back and discuss those specific topics that Luca was talking about. But if we do, in fact, find an intelligent agent behind the origin of life, I don't think the research will stop. It will not halt the chemistry in any way. People will still study the cell. Also, I do think the environments that Luca is mentioning, there's a lot more to it than he's actually saying. I would like to come back and get into more details on that, but I don't think that it's as simple as he's saying. I don't think we're getting anywhere close to this process. So it's just more wishful thinking and hope, just like he said. Thank you.
All right. I appreciate that concluding statement there. Tony and Luca, I appreciate the cordial debate, the intelligent debate on a very important topic. So that being said, let's give you guys just a minute or two break before we get into some of these solid questions. Any last minute questions, guys, in the chat, tag me. And if they are relevant to the topic, we can see if we can find some time to throw those in during the Q&A. So let me go over a few reminders for you guys. As I pointed out earlier, today was day 4 of a week of epic shows. So tomorrow we will be taking off, get some behind the scenes work done. If you check the upcoming live stream section, you could probably tell that March is packed. I've pretty well run out of dates in March to schedule some of these debate requests that are coming in. So we're now going to be moving debates into April. We're going to start filling up April and May, guys. So 2022 is a great year for debates, discussions, interviews. So Monday, we're going to be back here with Michael J. Ord, Mammoth's presentation. And later that week, we've also got Mark Drysdale back. The battle continues, Mark Drysdale and Dr. Kent Hovind. The total inability debate, CJ Cox and Dr. Leighton Flowers. That's going to be a great theology-related debate. We're also going to be having next month Tom Jump. This one's going to be epic. This will be huge. Dr. Kent Hovind and Tom Jump debating evolution. This is the evolution challenge we put out. We've been getting a ton of requests, and we have a ton of debates specifically on that topic for everybody. Luca will be back here next month as well, debating myself. So Luca, I'm looking forward to this one. Is there evidence for human evolution? So that's going to be fun. And the end of next month, we've got the big debate on justification. Sola fide, does the Bible teach salvation is by faith alone? Dr. Robert Sungenis and Kelly Powers, two powerhouses in the debate world. They will be debating this important topic. Also, Call Me Emo. He'll be here on March 1st. This is going to be a fun one as well. Is there reasonable evidence for evolution? Again, the Evolution Debate Challenge series continues probably until the rapture. Call Me Emo and Kent. And a good debate between two scientists, well-studied, well-educated, Professor David McQueen, Jason Torn, the Genesis flood debate. This one is, I believe, April 8th. April 8th. So just make sure you are checking the upcoming live stream section and the event section on our website, standingfortruthministries.com. Should be fully updated by now. Okay, Tony, Luca, break time is over, gentlemen. We are going to get into some of these questions. And as you guys know, whoever the question is for, we'll make sure we give you guys that person the last word. So if you guys wanted a little back and forth on some of these questions, go ahead. But that'll help us move along.
You know, Donnie, I am literally on fire. I'm not sleeping for, I think, 14 hours or so, and I cannot feel a thing.
Wow.
I can do it all night. Let's go.
No problem, Luca. We'll come back. We'll do it again.
You don't even look phased. Look at you. What's the secret? What's the secret? Coffee? What's the secret to no sleep, Luca?
Lots of Italian donuts, Luca, right?
Donuts.
That's why we call him the Italian Wonder.
Italian, there you go.
I do not.
And the Boston Brawler, Tony.
More or less.
So here's the first question that comes in from Jackson Rowe. Jackson Rowe himself, who we actually just scheduled for round 5. Jackson Rowe and Kent, they'll be debating on March 7th or 17th, I believe. Okay, so Jackson Rowe asks, what does Tony, so this one's for you, Tony. Yeah. Jackson's coming at you. Jackson asks, what does Tony think of tolins and amino acids forming on Titan?
I'm not exactly sure, but are we talking about the one of the largest proteins? Is that what he's mentioning?
I think Titan
planet,
a moon,
planet, right?
Yeah. Jackson, if you want to give,
yeah, I would say it has all the same problems: the racemic problem, the handedness, the right and left and handed amino acids. I don't think that's on its way to being solved. And some of the larger proteins, it's massive problem. We can find amino acids all over. I think they found amino acids back in 1969. There was a meteorite that landed. I think it was an 80-20 mixture. That's the best they found, I believe. So.
Yeah, Jackson clarified. Yeah, Titan's a moon. Okay, well, I appreciate that, Tony, my man. Cool name for a moon, Titan. So, Luca, we're going to hand it over to you.
It was in my presentation, so Jackson did bring up a point of mine. It's important to have those kind of compounds on Titan, and maybe we will even observe something similar to abiogenesis. Why not? We can, and sadly, I do not think I will be able to see that, but I think that sooner or later we will colonize Titan, because it's one of the easiest places to do so. So it will be very fascinating to go there and study the atmosphere, what's happening there. It's even possible that life of some form does already exist there. So it will be amazing to be there and study those kind of things.
All right, I appreciate that response there, Luca. Tony, if you want, you can have a quick final word. Question was for you.
Yeah, I couldn't hear him too good, but go ahead. I just
All
can't
right.
hear. I can barely read that or hear you.
I guess you're on your phone, eh?
Yeah,
Okay.
I can barely. I'm sorry. Go ahead.
No worries, Tony. So next question comes in from Redefine Living. He says, question for both. So we'll get you both in on this one. We'll start with you, Luca, since Tony started with the last question. So he asks, are you familiar with the recent findings in the Miller experiment that showed contamination from the flask affected the results? Luca, we'll start with you.
No, I'm not, but Miller experiment was quite hold, even though they think that can be relevant again in some areas. So yeah, but even if it was contaminated, I think we can still use what we know from back then. Experiments are always useful, even when they go wrong. So yeah.
Go ahead, Tony.
I would totally disagree. That experiment was a disaster. It didn't show anything. There were lots of acids and other garbage found that Miller produced. I haven't read any recent papers on it, but yeah, definitely want to catch up on that. I don't think it showed anything. They don't even use those type of flask experiments anymore because they're so, it just produces so much garbage.
I appreciate the responses and answers from the both of you, Luca and Tony. So next question comes in from Michael Knighton. $5 super chat. Michael, I appreciate the support. Guys, you're the life and blood of this channel. I really appreciate it. So his question is for you, Luca. He asks, so you're saying that the more we get advanced, that we'll find out something that happened primitively. So Luca, do you see a contradiction in that at all, Luca? I guess is his question.
Because if you look into anything like quantum physics or the physics of the atom, the atomic bomb, and so on and so forth, it's not like those processes were discovered just now, and it's a huge thing. It's just that when you study something, you will discover more. I can see no contradiction there. Quantum physics was as real back then than now, but it was just discovered in a century ago or so. So yeah, it's not like that. It does not work like that. You will find new things and you will understand them better, but I cannot see any contradiction in that.
Thank you, Luca, and thank you for the question and super chat. Michael, Tony, anything you wanted to add to that? Any points you wanted to make?
I would say the total opposite, that the only way we're showing advancements is with intelligent agency and interfering in all this chemistry, but it's probably in one ear and out the other with the YouTube atheist.
All right, I appreciate that. Tony, Luca, question was for you. So to be fair, get the final word. Go ahead.
I have nothing to say. Let's say that you do not need intelligence to get to some kind of reaction. Chemical reaction do happen all the time without any intelligence needed.
All right, thank you, Luca and Tony. Next question.
Sorry, go.
You sure?
No. I was looking to Bubblegum asking for the papers. I did put those in the chat. So
yes, I see them. Luca, do you want me to put those in the description box of the debate?
Yes, if you want to.
Yeah, definitely. Okay, so next question comes in from Brandon. So Brandon, I appreciate it. Brandon, our hardworking thumbnail expert. So, Brandon, thanks so much, brother. So, he asks, question for Tony. Do you think if they were able to produce life in a lab, that shows it's possible in nature or only possible via intelligence?
I would say intelligence. I mean, that would be the only way that they're actually pulling it off, along with all their, pure chemicals and designed equipment and all their controlled processes. That always gets ignored.
All right, I appreciate it. Tony the man. Luca, over to you.
Okay,
you
you need intelligence to understand the processes, but it's not like you cannot get to a point where you could bring up a scenario where those things can happen without intelligence, like spatial relativity and so on and so forth. They are really complicated, but they can do just fine without us. And that's the same thing.
Thank you, Luca. Tony, question was for you if you wanted a final word on that one.
Yeah, I think we're good. I mean, it's pretty much, if we showed life in the lab, that would prove that it took intelligence to actually do it and under very controlled environments. But yeah, we could, let me know when they actually pull it off though, Luca.
All right, Luca, Tony, great job so far. So next question comes in from C Science Film Labs. And his question is for Luca. He asks, what determines if a cell is dead? What is taken from or given to a cell that determines if it's alive or not?
The process inside it, when the processes like respiration and other protein production, so on and so forth, stops, the cell is dead. I do not think that there is an actual difference in the materials itself before and after death. It's just that the cell is not longer working.
Thank you, Luca. I appreciate the response. I appreciate the question from C Science Film Labs. Tony, over to you. Anything you wanted to add, points you wanted to make?
I think that's the $10 million question is how do we get it to be alive and reproduce? Nobody's ever come close to that. And what they're trying to pass off as life is nowhere near life.
Tony, I appreciate it. Luca, question was for you. Get the final word.
Yes, we can understand what a cell does pretty well. I can say that we do know in the case of the eukaryotic cell almost everything about it. It's not the same to be able to reproduce the cell because it's a very complex thing. It's not that simple, but we can understand basically anything inside it. I did study biology introduction. Okay, it was just a class to get some credits at my university, but we can understand basically any process inside it. It's just chemistry and physics. It's nothing else. And we do know basically anything about the cell.
All right. I appreciate the responses, gentlemen. Next question comes in from Power Word. I appreciate the question, Power Word. He says, question for Luca. If RNA is like a DVD of the code of life, and copying that code is the key for life to continue and reproduce, how can the DVD create the DVD player through unguided means?
Well, it's not like they are the same thing. If you are talking about DNA, you can assemble DNA from RNA. You just need to have the right conditions to do so. The bases are more or less the same. It just changes the shape behind it. So yeah, it's chemistry. It's I won't say that's simple. DNA is basically impossible to synthesize in nature, but it's not that impossible once you get to RNA. And that's a huge step. It's not that simple to get to RNA. But once you get there, yeah, you can get to DNA.
Thank you, Luca. And Tony, go ahead. Floor is yours.
I would say it shows all the hallmarks of design. Nobody has produced a cell, and nobody's even coming close. I listed a couple problems with RNA world, even by experts that are on Luca's side. So yeah, I don't think it's making any real advancements in a controlled laboratory environment. Yes, in a prebiotic world, I think it's plagued with all the same problems.
Thank you, Tony. And Luca, question was for you.
I will stand on my words. I do not think that's the same thing. Chemistry is very complex. It's not like we do know anything about it, but does not work as a machine. It's quite a different thing. We can observe very complex reaction happening right now in nature. It's not like it's impossible to do so. If you want a cool example, we do have compounds similar to organic ones, but made from silica and nitrogen. It's incredible. They cannot get that long as organic ones, but they are basically the same thing.
Luca, thank you very much. And gentlemen, we've made it to our final question. Great debate. Question comes in from Trucker. I appreciate the question. So he asks, question, at what point does mineral gain enough sensual awareness to recognize self-preservation? I think that's for you, Luca.
Mm-hmm. Mineral itself, never. But we were talking about a cell. It was not a cell, a protocell, with the exterior made by inorganic material, but the inside was still in organic material. So it's not like you have an inorganic organism. That would be really, really cool. But as for now, silica cannot get that far. It can get to compounds very, very close to the organic ones, but they cannot get that far because the bond it's not as strong as the one with carbon. The double bond, it's a quite complex things to explain. So yeah, let's say that.
Thank you, Luca, and also thank you to Trucker for the question. Over to you, Tony. Any thoughts, any points you wanted to make?
I don't think minerals or crystals are able to store code or generate code, but we can come back and have that discussion.
All right. Thank you, Tony. And quick final word there, Luca.
Yeah, you can store information in crystals. I think that they are trying to make computers out of it. So yeah, depends on what you mean when you say information.
Maybe more like a pattern in nature, but I think if it was done, that would be the direct result of an intelligent agent. But thanks for having me.
My pleasure. I appreciate it. Luca and Tony, you guys made for a fantastic debate. Very cordial, very sophisticated. Michael, the Canadian atheist, said that he sent in a question. And I'm just waiting for him to send it in one more time. I didn't see it, which means it may not have. I've been looking for it. It may not have made its way through, Michael. So if you did want to sneak it in now, let me see here. Okay, so we'll end it with this one. Michael, the Canadian atheist, I appreciate the question. Looks like it is for you, Tony. So he says, we've attributed lots to gods in the past, good harvest, fertility, etc. When we solve origin of life, where will God hide then? Tony, what are your thoughts on that, brother?
I would say just the opposite. It's the abiogenesis process that is always hiding in some environment we could never demonstrate. But the debate wasn't on God or the supernatural. I would just turn the question right back around on him. Once we produce life in the lab and it's clearly shown that an intelligent agent is involved, I mean, how would that prove that natural processes would ever do it?
All right. I appreciate that response there, Tony. And the question, Michael. Luca, over to you. Anything you wanted to add?
Okay. Let's say that I think that abiogenesis is not a problem for a god, per se. When I think about abiogenesis, that will be not a problem, because even a god needs some form of abiogenesis to make life. Life, even in the Bible, was not already there. It was made from non-living material. So abiogenesis is needed in either case. You just arguing for a god or not. So if you show abiogenesis, people with faith will just say, okay, God did it, and go on with their life, for me.
No, Luca. Abiogenesis and an intelligent agent producing life are two very different things. You believe it happened. Well, yeah. The YouTube atheist believes that it happened without an intelligent agency. They're very different.
I think that you have the process and God too. You can, if you want to believe anyway, you can just say God directed that process, and you
set up the parameters. Right.
All right, guys. Thank you for keeping it fun. Thank you for keeping it engaging. We got through all the questions. So great job, Luca and Tony. I really appreciate this debate. Tons of fun. Lots of great feedback in the chat. Before we close it down, though, as we always do, Luca, Tony, I want to give you guys some final words, final thoughts. Thank you again for giving us your time for this important debate. Why don't we start with you, Luca? Final thoughts, final words?
I had fun. I saw some comments in the chat that for them I was like a little bit not very excited, but I am. It's just that I never show these kind of things. So I had fun. It was really good for me, and I was waiting for this debate for quite some time. And I have to thank you to making this possible. And so thank you, Donnie. And I want to be back soon, and I cannot wait for our debate.
I appreciate that, Luca. I am looking forward to our debate as well. It's definitely going to be a lot of fun. And you and Tony, yes, this has been a much anticipated debate. You guys have been going back and forth for a while. So this was the battle. This was the war. And you guys kept it cordial and respectful. So great job to the both of you. Mitchell, thank you for the super chat. I appreciate all the support, all the super stickers and super chats tonight, guys. God bless you. I appreciate that. Okay, Tony, we're going to hand it over to you. Final thoughts, final words, and thank you for doing this.
Yeah, thanks for having me. I think Luca has already made up his mind, but this was the very topic that got me to change my mind. I was an atheist most of my life, but I think I made some reasonable points on some of the problems, some of the controlled conditions, and we can come back and do it again on some of those specific points that Luca was commenting on. And yeah, had a lot of fun, and let's do it again. Thanks for having me.
My pleasure. My pleasure. If it wasn't for you guys being so generous with your time, to all the debaters that show up as guests on this channel, we wouldn't have so many epic debates. So last thing I want to say before I let you gentlemen get out of here is after show, I believe, on Logical, Plausible, Probable's channel. I think it is being moved to later tonight. So between 1 and 2 hours from now, guys. So just check into John's channel. And you'll see if that after show is still going to be taking place. So I got a couple announcements as well. So Tony, Luca, I'm going to let you gentlemen get out of here. I appreciate you guys being willing to engage this debate. God bless you as well.
God bless, guys. And Luca too.
See you later, gentlemen.
Pleasure.
Thank you.
So,
all righty, there we go. Another debate in the books. I think we're close to 160 now. So anybody who's new to this channel, if you're not yet subscribed, make sure to hit that subscribe button. Share around this content because the truth is important. And on Standing for Truth, we do believe in critical thinking skills. It's why we hold debates alongside all the interviews and discussions and videos that we put out. So before I shut it down, though, I just wanted to go over a couple announcements. We went over some announcements and reminders a couple times during the debate, but I did just recently set some new shows that people might not have seen yet. So we're going to be having Dr. Ken Colson. He's going to be back here in March. I believe it's March 16th. So look out for that in the event section. Dinosaur soft tissue. So this is a huge topic. Huge topic. We just had Dr. Mark Armitage here, where Matt and I spent a great deal of time and study leading up to that interview. Just kind of looking into the best possible criticisms, rebuttals, objections from the skeptics and critics. And Dr. Mark Armitage, he decimated him. He dismantled him. You know, sorry to the old Earth creationists, but it's a great time to be a young Earth creationist. So definitely check that out. It's got the critics running. It's got the skeptics running. No rebuttal, no response, because there is no rebuttal and there is no response to the amazing, the fascinating evidence available when it comes to a dinosaur soft tissue and these biomaterials being found that are amazingly preserved. So please check that out. And again, we're going to have another show on that next month. Dr. Ken Colson, PhD geologist. He's done a ton of study on this topic. We've had him on in the past, I think about 6 months ago. So he will be back. Mike Ord, he's on frequently. I think he's been on the channel about 6 times now. Every time he's a blessing. He gives some fantastic lectures, presentations, and we always do a live Q&A. We do it because, as I've pointed out, critical thinking is important. So if you guys have questions, if there are skeptics out there that have objections, bring them. Tag me in the live chat, because when we have the experts on, we want you guys to have that opportunity to ask your questions, and we like to make our shows engaging and interactive. We've also got Christopher Silvius and Jackson Rowe. They will be here. The Great Transitional Fossils debate, that is on in March as well. Originally scheduled for March 15th, but I think we're going to move it to March 12th. Saturday, March 12th. Jackson Rowe, if you're okay with that, I see in the chat. I'm just going to try and space these out a bit. Man, March is, I think March we've been doing this now for a few years, and this March, next month, is the biggest month I think we've ever had for debates. I mean, the last couple days I've had a hard time fitting a couple of these in, so it is going to be huge. So I just want to space them out just slightly, and at this point, we're going to have to start scheduling for April, which we've already got a couple debates scheduled for April. So, guys, we're working full time for you. If you want to support us, please check us out on Patreon. You can support us over the standingfortruthministries.com. Without the amazing support that we get here on Standing for Truth Ministries, I don't know where we'd be. We definitely wouldn't be able to host all of these lectures and presentations with the top experts, as well as put on so many big debates, theology, science, philosophy, so on and so forth. So I appreciate all the support. This is a listener supported ministry. So God bless you guys. This one's going to be fun. Just scheduled this, I believe, March 31st. So this will be the final debate in March. Kent, he's a busy man. I think we've got about six debates for Dr. Dino in March. And January and this month have been no different. We've had about a debate a week, and they've always been epic. We're actually about to hit 1 million views on the channel, guys. We're at 950,000. So stay tuned. We're going to hit that 1 million view mark soon. If we can fit it in, we can have like a 1 million view party, open mic, if there's a day we can fit that in. Matt and I have also been working on a very thorough response to one of Gutsick Gibbons' videos on human evolution. That's going to be awesome. You guys are going to love that. We just got to find a day to fit that in, and we're going to do, we might make it a 3-part series. So yeah, definitely stay tuned for that. And yeah, that's it for now for new events. Earlier in the show, I went over another dozen events that we have for March and April. Again, guys, check the website if you're interested in a debate, or you have any suggestions or recommendations or questions. Feel free to shoot me an email, and I'll get to you as soon as I can. So again, Luca, I see you in the chat. Tony, great job tonight. Fantastic debate. The great Abiogenesis debate. And it really was a very intelligent debate. The discussion was great, and it's an important topic. So I believe, again, in the next couple hours, there will be an after show on Logical, Plausible, Probables channel. So look out there. If you're not subscribed to his channel, make sure to subscribe, because he puts on all these epic after shows, and he's been doing an after show pretty well for every single debate in the Evolution debate series. And we've got a ton over the next couple months when it comes to the Evolution debate challenge series. So he's going to be having a lot of after shows. Last night was a ton of fun on his after show. So make sure you subscribe to him. Also, our secondary channel, Young Earth Creation, where we re-upload a lot of our debates here, mainly the like science, age of the earth, creation, evolution type debates, not really the theology debates. So subscribe there. That's our secondary channel, and we're up to about 20,000 subscribers there as well. It was a blessing. We got a Noah's Ark video that we put out about a year ago, went viral. I think it's at about 1.3 million views. So just make sure you're subscribed to there as well. And Michael Hu says, awesome debate. Good night, guys. Good night as well. Everybody, thank you so much for tuning in. Tons of fun. Thank you for all the questions. You guys in the chat are awesome. The chats are always lively, and it makes the debate just that much more fun. And before I forget, check the website. We've fully updated it. Actually, it'll be updated again tomorrow. I'm having the website designer add our last 2 shows with Russ Miller and Dr. Mark Armitage. Those will be added to the homepage, because those are 2 must-watch shows. I mean, the evidence, the presentations, the information in those 2 videos are just so important. They are must-watch shows. So those will be added onto the website as well. And I think that's it. That's really all that comes to mind. So God bless everybody. Thanks again for tuning in to this great debate. And we'll see you all again on Monday for a presentation with Michael J. Ord on mammoths.